My name is Selina Wang. I am an associate professor of Cooperative Expansion and vice chair of the Department of Food Science and Technology here at UC Davis. My lab is an analytical chemistry lab that have been focusing on food quality, purity, and nutrition density. We primarily focus on 4 different areas. The first one is to identify important chemical markers for quality, purity, and nutrient density of foods. And that may look very different depending on the different crops that we look at. I started in olives and been studying olives and olive oil for the last 15 years. And we have also expanded to avocados, avocado oil, almonds, walnuts, pistachios, tomatoes, and other crops. The second area my lab works on is to develop robust, meaning faster, better, and cheaper detection methods that can be easily adopted by the industry. Many of the chemical methods that we use in academia, they can be very expensive and difficult to operate. So we do a lot of work to develop faster, better, and cheaper methods. So they can be as good as the traditional methods that are being used in academic labs, but they can be used in QA or QC for companies. One example is to develop faster and better, cheaper methods to detect rancidity for walnuts. The traditional methods using gas chromatography can be really difficult to use. So we have developed a benchtop method using rancidity, which is an accelerated aging test that can tell you about the stability and predict shelf life of the product. The third area my lab works on is to modify processing methods to improve quality, safety, and nutrient density of particular food products. This also includes to mitigate some of the contaminants or potential toxins that can happen during processing. One example is acrylamide is a known carcinogen that can be formed under high thermal conditions. For example, a retort process. And we have done some work to figure out that by reducing temperature slightly, you can maintain the safety of the product, as well as to reduce or completely mitigate the occurrence of acrylamide. The fourth research area that we've been working on, and especially in the last 5 years or so, is valorization of agricultural byproduct to increase crop value and sustainability. In particular, my lab has been working on this for olive pomace since 2012. We realized that most of the raw material actually goes into the waste stream during olive oil processing. So there's a lot of value in there in terms of mass, but also a lot of antioxidants, phenolics. They're more water soluble, so they are in the waste stream. So a lot of work has gone into extract the antioxidant, the phenolics, out of the waste stream and finding high value uses for them. One of the current research area for olive pomace in our lab is to figuring out how to use these olive pomace extracts as a natural sanitizing agent that can be used during pre-harvest, during harvest, post-harvest, as well as used as food preservatives to food products, and to reduce our need for synthetic preservatives, which is something that the consumers are wanting from food industries. So my lab tried to cover each step of the food system when we can, because every step counts to increase quality and nutrient density, and has an opportunity to improve sustainability for planetary and human health. So we are looking forward to work together with the Postharvest Center. You can follow our work at ucfoodquality.ucdavis.edu.